Hello everyone, my name is Bichthuy, and today on the topic of cancer, I will talk about cisplatin, which is an alkylating agent that inhibits DNA function by chemically cross-linking the DNA double helix. It belongs to one of the platinum complexes that are often used in therapy of cancer with the aim to induce tumor cell death. Here are some mechanism of action of cisplatin, which induces cytotoxicity by interfering with transcription and or DNA replication mechanism. Depends on the cell type and concentration. It has the ability to cross-link with the purine bases on the DNA. The first step begins after cisplatin penetrates through the cell membrane to replace one chloride ion from the complex with a water molecule. From there, the structure can bind to a single nitrogen on the DNA nucleotide. Studies suggest that cisplatin usually binds with DNA prefer nitrogen 7 onto adjacent guanine of the same strand. Cisplatin cross-links DNA, interfere with cell division by mitosis, and interfere with the repair mechanism that causes DNA damage, which in turn activate apoptosis when repair cannot be done. One figure in the book that I found related to this mechanism is from chapter 24, figure 24-28, induction of apoptosis by cell death signal. It began when the ligand binds to a death receptor on the surface of a target cell, and ends with DNA damage can also lead to apoptosis through the activity of the p53 protein, which p53 protein also involved in tumor damage via induction of apoptosis in cisplatin. Cisplatin is administered to cancer patients by uptake steroid saline solution through IV. Once inside the bloodstream, it remains intact due to high concentration of chloride ions. Cisplatin enters the cell by diffusion via specific receptors. There are many pathways that cisplatin can coordinate once it's inside the cell, and it has possible targets include DNA, RNA, sulfur-containing enzyme, and mitochondria, with the major one being DNA. Once inside the cell, cisplatin coordinates to DNA through nitrogen atoms of the DNA base pair, especially the number 7 atoms of purine. These nitrogen atoms are free to coordinate to cisplatin because they cannot make hydrogen bonds with other DNA bases. From there, form adducts, which is a cisplatin DNA complex product. One important adduct that mostly referred to due to its anti-cancer activity is the 1,2 intrastrand adducts, in which 2 chloride ligands of the cisplatin are replaced by purine atoms on adjacent bases on the same strand of DNA. When cisplatin binds to DNA, it affects both replication and transcription of DNA, as well as mechanism of DNA repair. It stops polymerases from doing the job. Cisplatin is super effective against testicular, ovarian, head and neck, bladder, cervical, as well as some cell lung cancer. Aside from that, some side effects of it include bone marrow suppression, which is the decrease of blood cells and platelets, nausea, vomiting, or even tumor resistance. One major side effect from cisplatin is toxicity, which can be varied from neurotoxicity, ototoxicity, which is hearing loss, and nephrotoxicity, which is kidney toxicity, that are usually dose related. This is what the toxicity occur from cells' inability to repair DNA damage, and mismatched repair activity can cause cisplatin resistance. So here's a question that I have. DNA researchers have conducted in-depth studies about possibilities that cisplatin cytotoxicity activity may come from the failure of the excision repair system. What do you think about this hypothesis? Thank you for watching.